Hello, everyone, and welcome to OHF's On the Road Summit webinar series. And today's session is going to be focusing on approved treatments and drugs in the pipeline. And I'd just like to welcome everyone. The goals today are we really want you to learn, to engage, and connect with our families, patients, caregivers, and our industry partners. And we're trying to build unity, and we want to activate our hyperoxaluria community. And this series is a unique opportunity to learn about clinical trials, drug development, financial assistance, and to interact again with patients, their families, research and drug development professionals around the world. So today's topic and the goals of this webinar, again, are to look at the approved treatment and drugs in the pipeline. And what we're going to do is give a brief overview of the science behind the promising therapeutic technologies that are changing the face of the primary hyperoxaluria and enteric hyperoxaluria treatment landscape. And the goal will be to do this at what I would call the 30,000-foot level, just to give you, again, an overview of the main concepts behind these therapies that are either approved or in development. I think it's important here to state again that OHF is a patient advocacy group, and because of this, we cannot favor one particular company or drug or strategy over another. And so what we're going to do is, again, just give an overview, and you will not see any drug names or company names on the slides. At the end, however, I do have a list of all of the companies that are kind of representing the different technologies that we're going to be discussing today. And I encourage you to go to the company websites to get even more detail on these different technologies, and also to look at the different companies' clinical trial plans. I'd also like to remind, before we get started, remind the patients and their families that science and drug development is a constantly evolving process. And as you may have heard the term personalized medicine, this is all of these technologies now are, I should say therapies in general, are moving towards what we call personalized medicine, where each treatment is being tailored to the particular disease. And we'll see today that they're being tailored to different forms of primary hyperoxaluria. And the goal is to have, obviously, a better quality of life. Again, I would like to encourage the patients and families to visit the company website to learn more about these technologies. And this will help you to be able to make informed decisions as you go forward. At the end of the presentation today, we will be hearing, we'll be hearing about Oxlumo from Alnylam, and we'll be hearing about the Alnylam Assist Patient Services Program. And before we begin, I also like to thank our sponsors and partners for their financial support and encouragement in their hard work to develop these different therapies. So my name is Todd Lowther. I'm a professor in the Department of Biochemistry and Center for Structural Biology at Wake Forest School of Medicine. And I'm the chair of the Scientific Advisory Board at the Oxalosis and Hyperoxaluria Foundation. Other speakers today will include Dr. John Liskey at the Mayo Clinic and Joanna Pearson from Alnylam Pharmaceuticals. So just some quick webinar housekeeping. This meeting will be approximately 1 hour. All the participants are muted to allow the speakers to present without interruptions, which is kind of a strange sensation of giving a seminar and having no feedback at all. You're just talking to a screen. This webinar is being broadcast live on Facebook, and it's also being recorded so that you can look at it later. If you have questions during the webinar, please put your questions in the Q&A box at the bottom of your screen, and we will answer them at the end of the webinar. And if we run out of time, we will address the questions by email. So again, I'm a professor at Wake Forest, and I just thought I'd say just 2 seconds about what's the Scientific Advisory Board. So this is a panel of basic science researchers and clinicians that advise the board of directors of OHF on a whole variety of different things, but in particular, we review grant applications for pilot studies and science projects for NPH and enteric hyperoxaluria. So the outline today, we're going to be, just so we're all on the same page, we're going to go over definitions of primary and enteric hyperoxaluria. I'd like to go over a quick biology 101 of what the function of DNA, mRNA, and proteins. We'll be looking at some genetic strategies for treating pH, small molecule drug strategies for treating pH, and then we'll move into thinking about therapies for degrading oxalate in enteric hyperoxaluria, and then we'll hear a presentation from Anilum on Oxlumo. So primary hyperoxaluria, or PH, is a rare genetic liver disease that causes the production of too much oxalate, and that oxalate goes into the kidney and causes kidney damage. The PH is caused by mutations in 3 enzymes that are involved in glyoxylate and hydroxyproline metabolism. The first that was discovered is alanine glyoxylate aminotransferase, or AGT, and this is found in PH1 patients. Glyoxylate reductase, or GR, is found in PH2 patients, and 4-hydroxyglutarate aldolase, or HOGA, is found. Those mutations are found in PH3 patients. Enteric hyperoxaluria, and I should say that again, this is a liver, these are liver enzyme defects which cause the production of too much oxalate. In contrast, enteric hyperoxaluria is a disorder where you are absorbing too much oxalate from the gut from the diet, and this can be caused by a variety of different things. And Dr. Liskey will be going over that in detail. So just a quick overview of what is DNA, mRNA, and proteins. So DNA is our genetic code. It's in the nucleus of our cells, and it's essentially the instruction book, or you can think of it as the hard drive of your computer, and it's containing the information that is needed to ultimately generate what we call proteins, and we'll talk about that more in a second. The intermediate step is to convert the DNA into what we call RNA, and this RNA is what we call the messenger, and it's essentially the information string which takes the code from the nucleus into what we call the cytoplasm, or the outer compartment of the cell. And this information string, again, is thinking about your computer, is basically like a series of ones and zeros in a computer program that's telling the program what to do. In this case, we have RNA bases, which are a particular type of biological repeat unit that are made, that are read and made into a protein. And so the protein is the final product, and it's the genetic code tells the cell what type of protein to make. And depending on the protein, it will have a particular 3-dimensional structure. And from that 3-dimensional structure, the protein will have a particular job within the cell. And you may relate a little bit more to what a protein is, because the proteins are made of amino acids. So if you take like a protein bar, or you have a protein shake, those are basically amino acids that are in the free form. But in a protein, they're linked together in a chain, they fold up, and then that causes them to have a particular function or job within the cell. So one of the functions could be that these proteins are part of the structural components of the cell, basically the architecture of the cell. But for today's webinar, we're going to be focusing more on enzymes. And enzymes are proteins that perform a particular chemical reaction. And the reason why this is important is that under normal conditions, the cell is doing all these different reactions. But when we have a mutation in the DNA, as we do in PH1, 2, and 3, that mutation changes the protein, and then it loses or has an altered function. And because of that altered function, the metabolism has changed, and now we're making too much glyoxylate and too much oxalate, leading to the formation of kidney stones. So the first genetic strategy that we're going to look at is called genome editing, and we're going to basically use scissors as a metaphor for this. So again, we're operating, these technologies are operating at the DNA level. And so the concept here is that we just want to correct the gene. So if we have a mutation in AGT, for example, in this piece of pink DNA, we're going to cut that DNA out with scissors. And the scissors that we're going to use are called nucleases. These are indicated over here. The technical name for some of these nuclease scissors are indicated here. But again, the concept is that we're going to cut this piece of DNA out, and then we're going to replace it with a repaired copy. And so now you have corrected the gene, and now the mutated or misspelled gene is now going to be corrected and will make the correct protein, and the protein will have its intended, originally intended function. Some of the technologies that companies are focusing on are not correcting the gene, but they're deleting another gene or another, and in that case, a protein that basically is involved in the formation of glyoxylate or oxalate. So if we remove these genes, then we block the formation of glyoxylate and oxalate. So again, the two strategies that we're going to look at is we're going to look at correcting or replacing the defective AGT, GR, or HOGA genes, or we're going to delete genes that promote oxalate formation. And one of the examples of this is glycolate oxidase, GO, in pH1. So another strategy that we could use for treating pH is to destroy the mRNA of the target protein. And this is like turning off a light switch. So in the last slide, we saw that we can change the DNA to correct the protein. In this case, though, we're going to knock out the production of a protein by turning off the light switch and destroying the RNA. And the way this happens is that the drug is called a small interfering RNA. This is the drug. When you inject the drug into the cells, it's going to bind to what's called the RISC complex. And when it forms that complex, the siRNA and the RISC complex is going to bind to the target mRNA, and then it's going to cause it to be degraded. And because the mRNA is degraded, you no longer make the protein. And so two targets that we'll talk about are glycolate oxidase and pH1. So if you remove glycolate oxidase, you can lower urinate oxalate levels. And if you remove lactate dehydrogenase, you can also lower urinate oxalate levels. So an alternative strategy would be to use what I would call a traditional small molecule approach. And so these are kind of what we're used to thinking about as a drug, is we have a pill that we take by mouth and we swallow it. And the concept here is that we want, now instead of working on the DNA or the RNA level, we're directly going to work on the protein level, and we're going to block its function with this drug. So in order to understand how this works, we need to understand how enzymes work a little bit. So an enzyme, again, is a protein that performs a catalytic or a chemical reaction, and I've indicated the enzyme here as a little blue Pac-Man. And what happens is the molecule that it works on, which is called the substrate, binds to the active site. And the important thing here is that the shape of the substrate is important. And what you can see here is I have this diamond, and because of this particular shape and the shape of the enzyme surface, it can bind this diamond shape. Once it binds the diamond, then it can perform the chemistry and produce the product. And what you can see is that we've changed the molecule from the substrate to the product, which is this cyan blue sphere. So if we want to inhibit this normal process, we can develop a drug that binds to the enzyme. And that is what is shown here. So what we can do is we can design an inhibitor which mimics the shape of the substrate. So you can see basically I have the bottom of this diamond, and this portion of the molecule can bind into the active site or into this surface of the Pac-Man, and it blocks the ability of the substrate to bind. So this is basically putting on the brakes or putting on, holding up a stop sign and saying that now the enzyme can no longer make the product. And so one way you can think about this is if this was an enzyme like LDHA that makes oxalate, if you had an inhibitor that blocks LDHA, it would no longer be able to produce oxalate. So the two examples that we're going to look at right now are blocking GO and blocking LDHA. So again, we're going to be blocking the protein function by adding a small molecule glycolate oxidase inhibitor, and this would be specific for pH1. So again, we would be taking a small molecule inhibitor in a pill by mouth. When the drug goes into the body, it would reach the liver and block or inhibit the glycolate oxidase enzyme. In pH1 patients, we have, again, mutations in the AGT protein, and they're no longer, they're not able to convert glyoxylate into glycine. And because of that, they have excess glyoxylate. LDHA converts it to oxalate. But it's a little bit complicated, but there's another situation in the cell in that the glycolate is also being converted into glyoxylate. And it turns out that this is a predominant step. So if we have a GO inhibitor that blocks glycolate, that prevents glyoxylate production and also oxalate formation. And so again, the net result would be that by inhibiting GO, you're going to lower the amount of oxalate that's produced in the liver. In a similar way, we can block the function of LDHA with an inhibitor. So the inhibitor, again, would be in a pill form. It would be delivered through the bloodstream to the liver and block LDHA. The difference here is that LDHA is one step downstream, and it is actually the final step of converting glyoxylate to oxalate. And so blocking LDHA would be has the potential to be applicable to all three forms of pH, whereas GO is currently thought to be more applicable to pH1 patients. So I'm going to hand over the presentation now to John Liskey. He's a professor of medicine at Mayo Clinic, and he is the director of the OHF Primary Hyperoxaluria Registry and an expert in enteric hyperoxaluria.
There we go. So good morning, everybody, and good afternoon, as the case may be. I'm happy to be here and switch gears just a little bit and still talking about oxalate, but talking in this case about enteric hyperoxaluria. So next slide. So this is a schematic representation here. This is your colon. And it turns out that the oxalate that we get from our diet and the things that have oxalate in them are all plants. And they come in the form of calcium oxalate crystals that are inside of these plants, things like spinach or nuts or other things that have lower amounts of oxalate, but it's always calcium oxalate crystal that's inside that plant. And ordinarily, not so much of that gets dissolved or absorbed. So in a common ordinary circumstance, only about 10% of the oxalate you might eat in something would actually end up absorbed and have to go out into the urine. But in this circumstance, when it gets to the colon, if you have fat malabsorption, so there are many potential causes of this, where you're not handling fats, which should be absorbed almost entirely in your small intestine, they get to the colon, and in this case, they can bind up with calcium, and that changes the balance of things so that there's more calcium binding to these fats, and that in turn frees up more oxalate. And then this free oxalate can be absorbed more readily into the bloodstream. And so suddenly the amount of oxalate isn't 10% that you absorb from these things that you might eat, but becomes 20 or 30%. And the net result of that is then this oxalate has to go through the bloodstream, and then the only way the body has to get rid of that is to excrete it in the kidneys. That we don't, unfortunately, have an enzyme in our bodies that can break down oxalate. So this has to be eliminated, and it can lead to issues like kidney stones or kidney failure if it's quite high. There can be other factors here as well. So there can be increased permeability. It's thought that in some of these diseases that cause fat malabsorption, you also don't absorb your bile acids that come from your gallbladder. And these can be harmful to the lining of your colon. And so the permeability may increase. So that may make things even a little bit worse. But the net result of all of these is that you have too much oxalate around and it's getting into the bloodstream. In your intestine itself, and I'll start just focusing down here on human microbiome. So that's sort of a fancy word, but it turns out that your colon is not sterile. Nobody's colon is, and we have millions and millions of bacteria there. And it's a very complicated mix of bacteria, and they work together. And in health, it actually probably does good things for us. One of the things that they can potentially do is break down oxalate. So it's known there's one particular bacteria called Oxalobacter that is a well-known bacteria that can break down oxalate. But there are probably many, many other bacteria that can do that to some extent at least. And if they do that, then this free oxalate that's in your colon potentially will not ever get absorbed, but it's broken down to other molecules. In many conditions, there have been attempts over the recent decade or more to try to alter your microbiome in the intestine. And this typically involves probiotics. So many of you may have taken various probiotics that have what we consider to be good bacteria and try to reset the balance. For example, if people take antibiotics, sometimes your microbiome may get out of whack, and it's thought that in some circumstances, at least, these probiotics may help get the good bacteria back in the game. But more recently, there have been more, I would say, sophisticated attempts to engineer bacteria that might do good things for us, and in particular, looking at this in the world of oxalate biology. And so in this case, these are called synthetic biotics or engineered bacteroides. So that's just fancier words for what they're doing to try to make these bacteria. In this case, they're taking the enzymes that have been identified in these other bacteria like Oxalobacter, but putting them in other bacteria or other strategies to try to make a bacterial combination that if you took it by mouth would digest the oxalate, and then it would never have to be absorbed. And so this is an active area of investigation, and there are many entities looking at this. Okay, next slide. The other strategy that would be quite similar is to actually take the enzyme. So we know that, for example, oxalate decarboxylase is an enzyme that breaks down oxalate. And if you take enough of this in the right time, this could also break down the oxalate, and then you would never have to absorb it as well. So there are also many entities looking at this. There are different forms of this enzyme. There are a lot of issues that have to be overcome. How much do you give? What exact type of enzyme do you use? How does it work in the intestine versus the colon versus the stomach? And all of those aspects are being worked on, but this is there are actually studies ongoing with this as well, looking at these sort of oxalate decarboxylase enzymes, and can we use that effectively to break down the oxalate so that it never has to be dealt with by the kidneys. Okay, next slide. So I think that's it. So I'll just stop here and thank all of our sponsors here, particularly Alnylam and Dicerna at the top here that are helping to put this webinar on for us today, and for all these companies that are working with the OHF to move the field of oxalate research forward. I'd also now at this point like to introduce Joanna Pearson with Alnylam, who is going to talk a little bit about Oxlumo, which we're very excited is available now. It's FDA approved in the United States. And the disclaimer there, for all questions about Oxlumo, you should talk to your doctor. And for questions about the Alnylam Assist, you should contact the Alnylam Assist program. So thank you, Joanna.
Thank you so much, the OHF and everyone for being here this evening. This afternoon. My name is Joanna Pearson, and I'm a patient education liaison with Alnylam Pharmaceuticals. So if you can go to the next slide, please. So I am here briefly to talk to you a little bit about Oxlumo, and then about Alnylam Assist, which is a patient services program for patients prescribed Oxlumo. And I do have to just say, Oxlumo is a prescription medication for the treatment of primary hyperoxaluria type 1, also known as PH1, to lower oxalate in urine in children and in adults. So before I move on and start sharing more information about Oxlumo, I'd like to tell you how you can access our full prescribing information for Oxlumo. You can use your smartphone to scan the QR code that's in today's presentation, or you can visit www.oxlumo. And in addition, I have put a link in the to the prescribing information in the Zoom chat. So next slide, please. So how does Oxlumo work? So Oxlumo, or also known as lumasiran, targets oxalate production in the source. It works in the liver to reduce the amount of oxalate produced by the body, which is the cause of primary hyperoxaluria type 1, or PH1, the symptoms. So by reducing the oxalate, Oxlumo targets the cause of PH1 symptoms. Because Oxlumo reduces oxalate production, less of it circulates in the body. Next slide. Thank you. So I would like to read a little bit more about our important safety information for Oxlumo, or lumasiran. So the most common side effects of Oxlumo is injection site reaction. Redness, pain, itching, and swelling are the swelling at the injection site. These are not all the possible side effects of Oxlumo. Talk to your doctor about side effects that you experience. You are encouraged to report negative side effects of prescription drugs to the FDA. And you can do so by visiting the website at www.fda.gov/medwatch, or you can call the 1-800-FDA-1088. So Oxlumo has not been studied in pregnant or breastfeeding women. So talk to your doctor about the risk of taking Oxlumo if you are pregnant, or if you are pregnant, plan to become pregnant, are breastfeeding, or plan to breastfeed. And also, for additional information about Oxlumo, please scan the QR code that's available at the top right corner of the slides today to view the full prescribing information, or visit www.oxlumo.com. Next slide, please. So speaking with your doctor is the first step in finding out if Oxlumo or lumasiran is right for you. To learn more about getting dedicated support throughout your treatment with Oxlumo, you can visit alnylamassist.com. You can also call 1-833-256-2748 anytime between the hours of 8 a.m. and 6 p.m. Eastern Standard Time, and that's Monday through Friday. Next slide. Alnylam Assist services include a dedicated case manager and support team. Case managers can help you locate resources and answer questions about getting started on treatment with Oxlumo or lumasiran. Help navigating insurance benefits. Alnylam Assist can help you and your family understand your insurance with Oxlumo. And then there's financial assistance. Patients who qualify may be eligible for commercial copay assistance or other financial assistance programs. And then please note, individuals must meet specified criteria to qualify for assistance. Alnylam reserves the right to make eligibility determinations and to modify or discontinue the program at any time. Next slide. So here's how to get started with Alnylam Assist. Step 1, talk to your doctor about whether Oxlumo or lumasiran is right for you. Step 2, complete the start form with your doctor. Step 3, your doctor will then submit the start form. And step 4, a case manager will contact you to help you understand your insurance coverage and to explain support services available to you through Alnylam Assist. Next slide. So I just want to follow up with this slide. Things to remember about Oxlumo patient support programs. Talk to your doctor about whether Oxlumo is right for you. Alnylam Assist support services are here to help you throughout your treatment journey. Alnylam Assist team includes a patient services liaison or a PEL and a case manager. And when starting Oxlumo, your case manager will help you understand your insurance coverage for the product. So thank you so much for your time. That is my last slide. Thank you for your attention, and I will turn the program back over to John. Thank you.
Great. Thank you very much, Joanne. So just have a couple of slides here. I know we've got a nice audience here this afternoon, but how can you get involved? Well, we think it's really important that you make your voice known, that really the patients speak most loudly for any disease, and particularly a rare disease. So the OHF is your organization, so please get involved with the OHF. They can provide support and they can really put your case forward in a very effective way and have done so for many, many years now. You should learn about and participate in any clinical trials if at all possible. Since this is a rare disease or rare diseases we're talking about today, really the number of people that are out there that can be in the studies is relatively limited, so that we can only advance the science and the treatments if people actually are willing to be in the studies. Patient registries are quite important. This has really helped us understand primary hyperoxaluria and has really been a key part of why we've been able to get studies done and treatments through the regulatory process successfully. So that's really, really important. We are ongoing with that effort, as well as additional questionnaires. There is data that is not really in the medical record that we get directly from patients. So the OHF has registries planned for both PH and EH, and please participate if possible in those, because that will be very helpful for the field moving forward as well. So next slide. And there's one in particular that is just about to go live, hopefully within a week or two. We're doing a primary hyperoxaluria questionnaire. We want to understand what it's like for patients and caregivers that live with primary hyperoxaluria. What are the biggest problems that you face? What would you like to see change in the future? We really need as many people as possible to participate. It's designed to be relatively quick and easy. There will be some compensation for your time. This particular survey, we'll point out, is just for United States based patients and families at this time. The email is shown there, hyperoxaluria@mayo.edu, if you're interested, and just you can say I want in, or any sort of communication, and we'll be back in touch. And this will be done entirely electronically, so it should be pretty easy and seamless for you to participate in. So next slide. This is, I think, where we will field the questions that are out there.
That's great. Thank you, John and and I am looking now to see. So does that chat screen show up on my screen, John?
I think under Q&A, I see.
I see it. I'm just saying, I'm wondering if you see it when
I do.
Okay.
So I think the question I see here is, when will lumasiran be available in India? And I think that unfortunately, we do not have any information about that. I don't think that there is an answer for that at this point in time. I know that people, I'm sure, are working on that with Alnylam, but I think there are certainly hurdles to overcome. So unfortunately, we don't have any great information on that.
That's right. We also had some questions submitted beforehand, and one of these was, when will the technologies be available? And so, as we've just heard about Oxlumo, that particular therapy went through, a several-year process of being optimized and validated by testing, not only in animal models of pH, but also in PH patients through a clinical trial. So again, that's emphasizing the need to participate in clinical trials. So the other technologies that we talked about today, they're in different stages of development, but some of those will also be, if they haven't already, will be transitioning to the early clinical trial stages. And so then we will need patients and families to be willing to participate in those studies. And as John indicated, unless the patients participate, we won't know if the therapy will work. And again, if we don't have that data, we can't convince the FDA or the regulatory agencies in Europe and around the world to approve using these therapies. We need to prove the therapies need to be shown to be successful in lowering urinary oxalate and also proved to be safe. John, there was another question that came in that said, will all these different therapies be for children and adults?
So yeah, I think that it's complicated doing these kind of studies in children. So typically, the earliest studies are done in adults to make sure that a given treatment is safe and effective, and then sort of works its way down through adolescence and then to smaller children last. And so you may have seen this with the current COVID vaccines, that the trials were initially in adults and then in the school age kids and then in the smaller kids finally. And I think that's much the story and things that would happen here in primary enteric or enteric hyperoxaluria. And that was, the strategy with Oxlumo, that they started with adults, and then once it looked effective, then they moved down to the smaller children. So I would say, yes, that would be the goal for any of these treatments, but, we have to be very careful and make sure that things are safe before we do that.
That's great. And I just want to encourage you that if you have questions, you can paste them in the question and answer on the Zoom window or on the Facebook Live page. So another question that we get often is, where can I sign up for a clinical trial? So certainly you can contact the OHF, and we keep track of all of the ongoing clinical trials and can help direct you and make connections. But you can also go to clinicaltrials.gov, which is the U.S. government website that lists all of the ongoing clinical trials, and there's actually quite a bit of information there as well. And John, since you see patients that are on clinical studies, maybe you can answer the question, is it safe to be on a clinical study?
I'd say my opinion would be definitely yes. All of these kind of studies are very heavily regulated. Certainly, any place that you would go for a study, any medical center, there is a local institutional review board, or IRB, that will review the protocol, and other doctors and healthcare workers will look carefully at the protocol, and they really will question if they think that something isn't being done in a very safe manner. So I would say yes. And then the companies themselves want things to be safe and effective. And so if it is a trial for a treatment like we were talking about today, those things are reviewed by FDA. And so the companies are actually partnering with FDA or European agencies to design their studies and satisfy what might be considered safety concerns, so that the study ultimately will be as effective as possible. So I would say, yes, you can be very assured that everything's being done at all possible to make these things safe.
And John, since you're talking about patients, can you handle this question from a patient? So for patients coming into childbearing years, should there be concern about family life planning? I realize there is no information on medication for pregnant people, but what about freezing eggs or sperm before using the medication?
Yeah, so that's, I would say we don't really know the answer to the question about how safe, for example, the siRNA treatments, the Oxlumo that's been approved. if that what that impact that would or would not have on somebody who's pregnant or a partner of somebody that wants to get pregnant. So I would say, we don't know that it's not safe, but certainly doing something like freezing eggs or sperm would be a very reasonable thing to do, and that's done very routinely for other circumstances. So I think that's something ultimately you would want to talk to your physicians, perhaps your obstetrician, about what they would recommend in your particular case. I just think we don't really know the answers to those things yet.
Right. We also had a question, just wanting clarification. Both Nedosiran and Oxlumo are both, they wrote mRNA treatments. So they are technically called siRNA treatments. So that, again, the drug is going into the cells and using that RISC complex to degrade the mRNA of the target protein. So both Nedosiran and Oxlumo are using this siRNA type of approach where you're degrading the mRNA. Let's see. Very hard to look at 3 windows at the same time. I'd also just like to say, like, if you're looking at this webinar, recording, if you have questions, you can send them to the OHF email and we'll work to get those answered. And if you did not get your question answered today, we'll do the same thing.
So I see one here in the Q&A asking about PH1 patients on dialysis and the use of lumasiran. And yeah, I think those studies are ongoing. I think that we would hope that, certainly if it works the way that we think things should work, it should decrease your liver production of oxalate, and that should help lower the blood levels of oxalate for people that are on dialysis. But I would say that those studies are still ongoing. So we'll have to see it, but I think there will be some information quite soon on initial impressions with that. And then the follow-up question down here had to do with liver kidney transplant, and would people on dialysis still need to get a liver kidney transplant if they were being treated with something like Oxlumo? And again, I think that that's an exciting thing to think about, but we will have to see the data and see how well people respond, and what the best treatment will be, a year from now as opposed to what it was a year ago. So I think we're in an exciting time where things are changing quite quickly. So again, the jury's out.
That's right.
Here's another question. It says, Oxlumo stops new production of oxalates by suppressing liver enzymes. What happens to existing oxalates present inside the patient's body? Will they get dissolved? So I think that the answer to that would be no, that the Oxlumo really only turns off production in the liver. So there'll be less sort of oxalate going into your bloodstream, and then hopefully less oxalate actively depositing throughout your body. But it wouldn't directly cause any of those deposits, if they happen to be there in your bones or other tissues, to dissolve. On the other hand, if someone's on dialysis, oxalate is being removed with each dialysis session, or if somebody gets a kidney transplant, the oxalate then starts going out through the urine. So that we do know that those oxalate stores through the body, if you start actually removing it from the body, they'll change the fluxes, and it does get removed from the body over time. So people that have gotten, kidney liver transplants, we know that eventually the urine oxalate excretion goes down to normal, although that may take a year or two. So sort of a long answer, but I think that yes, this treatment could, with other things going on with it, potentially help to resolve oxalosis, but it's not a direct effect of the drug itself.
John, do you see any other questions there? I'm not seeing the screen you're looking at. So if you see any other ones there, that would be great.
It says, by taking Oxlumo, there's a third question. Shall we expect kidney to get reactivated to some extent, improvement in GFR level and urine? So that's another really good question. And we do know that in certain circumstances, if we can turn off the oxalate damage to the kidney and you still have kidney function left, that probably to some extent, some of those deposits in the kidney can be reabsorbed and some of the scarring can improve a little bit. But I would say there's a limit to that. So if somebody is in kidney failure and been on dialysis for any period of time, I think it's pretty unlikely that you would see kidney recovery. But if we happen to intervene right when people are getting to kidney failure or very quickly thereafter, if it's been a very fast thing, yes, I think there can sometimes be a small improvement, but we don't typically see it get, back to normal, say. So every case is a little bit different that way.
Okay, John, if you see one more question, let's take that, and then we'll, again, if we don't answer all the questions today, we'll follow up via email.
So it says, there is one more I see. It says, how does the OHF work with clinical trial sponsors and patients to advance research in PH1, and what are the biggest challenges to research today? Well, I would say that, the OHF is really works with the patients, and they serve as the patients' advocates and try to work with them and all companies and try to educate the patients about what's available, and then also educate the companies and sort of serve as a bridge between the two. So I think that where the OHF is very, doesn't really have an opinion about any company or technology, because we understand the OHF that we don't really necessarily know what the answers are going to be. We may think something looks really promising today and it turns out that that doesn't work, but something else really does work that we weren't really as keen on. So I think that really that's the approach of science in general, that we really have to be open to all possibilities. And the OHF has sponsored a lot of basic science research over the years. And I think that's really set the stage for the companies coming in, that if we hadn't, the OHF had not sort of made people understand what causes primary hyperoxaluria, what the enzymes are, what happens to patients, the companies would not have necessarily been drawn to this particular disease, which has really attracted a lot of attention in the last couple of years.
Thank you. So again, if you have additional questions, please continue to put them in or email them to the OHF email address. I'd like to thank John and Joanna again for being here today. Thank you again to our sponsors, in particular Alnylam and Dicerna. And also like to let you know that there will be an upcoming fall webinar series, and that's being decided and worked out right now. And so just check out ohf.org or the OHF social media pages for updated information. And we look forward to sharing additional information with you in the future. Thanks again for joining today, and all the speakers today. We're very happy to do it and to help educate everyone, in particular the patients and the families. Thank you.
Thank you.